Good morning, dear children. I hope you all are fine. I welcome you all to the second chapter in our syllabus Cell, the basic unit of life. Our earth is a beautiful place wherein different types of organisms happily coexist. From small masses to huge trees, and from small bacteria, invisible bacteria, to large human beings or blue whale, all the organisms are made up of a basic unit called cell. Before 350 years ago, that is before the discovery of microscope, people didn't know that there was a world which we could not see with our naked eyes. So after many scientists were observing and describing this unknown world with the help of magnifying glasses or microscopes. Among them, some of the scientists like Athanasius Kircher, Jan Swammerdam, Anton von Leeuwenhoek did many findings in the discovery of the unaware world which we could not see with our naked eyes. Anton von Leeuwenhoek was the first human being in 1674 to see living bodies like bacteria, yeast in a drop of water. He prepared several types of magnifying glasses and used his lenses to study both living and non-living things under the microscope. So whatever he found, he used to draw the sketches of the organisms like in the picture what you can see here. It was in the year 1665. Robert Hooke, a British scientist, observed thin slices of cork under a simple magnifying device. He observed that the cork resembled the structure of a honeycomb consisting of many empty spaces like structures. So by seeing that empty cavities, he named them as cell. So the meaning of the word cell in Latin is a little room. Here in the picture you can see the simple microscope used by Robert Hooke, and how did Robert Hooke observe the cork tissue. So you can see there are small cavities. As cork tissue is a dead cell, you can find just small cavities in the tissue. So that cavities were named as cell by Robert Hooke. Now let's try to understand what Robert Hooke had been observed in the dead cork tissue by performing a small activity to observe the cells in the matchstick. So for that, you need to soak the matchstick for a half an hour in water, and after that, as it becomes a little soft, you can cut the thin slices of that matchstick and place it on a slide. And you have to place a cover slip over that tissue and observe under the microscope. So while observing under the microscope, you need to draw the figure what you have observed. So here you can see the picture how the dead tissue, the cells in the dead tissue observed. So in this manner you can see the cells. The discovery of the cell was a major milestone in the history of science, and it gave the platform to still perform more activities in the functions going on inside the cell. So in the previous activity, you have observed how the cells look like in a dead matchstick tissue. But children, can you say that the cells in a dead tissue and the cells in a living tissue are same? So to find out that, we need to do another small activity, observing the onion peel cells. So for this, you need to peel out an onion, cut a small portion of inside part of the onion. And when you take the inside part of the onion, you find a small transparent membrane in the inner piece. So that transparent membrane you have to peel and place it on a glass slide and cover it with cover slip. So that inner membrane you can observe in this picture. So that slide you have to observe under the microscope. So after observing under the microscope, you need to draw the sketch of what you have observed. So as you can see in the picture here, you can observe the living plant cells. So there you can see a dark colored spots. Those are nothing but the nucleus which is present inside the cell. In this manner, you can observe the onion peel cells. These onion peel cells are plant cells. Now children, let us observe the cells from our own body. That is, observing the human cheek cells. So already you would have prepared the slide for onion peel cells. So now you need to prepare a slide to see your own cheek cells. So before that, you should wash your mouth cleanly. Take a clean wooden or plastic spoon and scrap the inner surface of your cheek. So while doing this, you need to keep two things in mind. Firstly, you have to wash the spoon properly before using it. And secondly, you should not scrap too hard. If you scrap too hard, you may injure yourself. So keeping these two things in mind, you need to collect the inner lining of your cheek and place it on a glass slide. After that, you need to cover it with a cover slip and observe under the microscope. So while you are observing in the microscope, you need to draw the sketch of what you have observed. So here you can see the picture, how you would observe the human cheek cells. And different parts of the cell you can see like nucleus, cell membrane and cytoplasm in the microscope. So can you notice any difference between the onion peel cells what we have seen in the previous activity and human cheek cells what you have observed in this activity? So children, just have a look of this picture where you can see onion cells under the microscope and human cheek cells under the microscope. So how do you know that this is an animal cell and this is a plant cell? What structures you can find while differentiating both the cells? So the major difference you can see is in the shape of the cells. The onion peel cells you can observe are in hexagonal shape or rectangular shape, but the human cheek cells will be in spherical or round shape. And still more small difference you can find here. The dark colored spot in the cell is to one side in a plant cell, and in the human cheek cells you can observe it in the middle of the cell. Like this, we can distinctify many differences between plant cells and animal cells. So what are the differences between plant cell and animal cell? And the next concept we can discuss in our next videos. This is the end of part 1. Thank you children.